Genetic engineering, also called genetic modification or genetic manipulation, is the direct manipulation of an organism's genes using biotechnology. It is a set of technologies used to change the genetic makeup of cells, including the transfer of genes within and across species boundaries to produce improved and novel organisms. A construct is usually created and used to insert this DNA into the host organism. The first recombinant DNA molecule was made by Paul Berg in 1972 by combining DNA from the monkey virus SV40 with the lambda virus. As well as inserting genes, the process can be used to remove or knock out genes. The new DNA can be inserted randomly, or targeted to a specific part of the genome. An organism that is generated through genetic engineering is considered to be genetically modified (GM), and the resulting entity is a genetically modified organism (GMO). Genetic engineering has been applied in numerous fields, including research, medicine, industrial biotechnology, and agriculture. In research, GMOs are used to study gene function and expression through loss of function, gain of function, tracking and expression experiments. The rise of commercialized genetically modified crops has provided economic benefit to farmers in many different countries, but has also been a source of most of the controversy surrounding the technology. Creating a GMO is a multi-step process. Genetic engineers must first choose what gene they wish to insert into the organism. The next step is to isolate the candidate gene. The cell containing the gene is opened and the DNA is purified. The gene is separated by using restriction enzymes to cut DNA into fragments, or polymerase chain reaction (PCR), to amplify up the gene segment. These segments can then be extracted through gel electrophoresis. Once isolated, the gene is ligated into a plasmid that is then inserted into a bacterium. The plasmid is replicated when the bacteria divide, ensuring unlimited copies of the gene are available. Before the gene is inserted into the target organism, it must be combined with other genetic elements. These include a promoter and terminator region, which initiate and end transcription. A selectable marker gene is added, which in most cases confers antibiotic resistance, so researchers can easily determine which cells have been successfully transformed. The gene can also be modified at this stage for better expression or effectiveness. These manipulations are carried out using recombinant DNA techniques, such as restriction digests, ligations, and molecular cloning. There are a number of techniques available for inserting the gene into the host genome. Some bacteria can naturally take up foreign DNA. This ability can be induced in other bacteria via stress, e.g. thermal or electric shock, which increases the cell membrane's permeability to DNA uptake, and DNA can either integrate with the genome or exist as extrachromosomal DNA. In plants, the DNA is often inserted using agrobacterium-mediated recombination. As only a single cell is transformed with genetic material, the organism must be regenerated from that single cell. In plants, this is accomplished through the use of tissue culture. In animals, it is necessary to ensure that the inserted DNA is present in the embryonic stem cells. Further testing using PCR, southern hybridization, and DNA sequencing is conducted to confirm that an organism contains the new gene. Critics have objected to the use of genetic engineering on several grounds, that include ethical, ecological, and economic concerns. Many of these concerns involve GM crops and whether food produced from them is safe, whether it should be labeled, and what impact growing them will have on the environment. These controversies have led to litigation, international trade disputes, and protests, and to restrictive regulation of commercial products in some countries.